Welcome to the second part on chapters 46 through 47. So we're going to do a quick run through. So anatomy for the human male. We're going to have the seminiferous tubules are going to be the main part of the testes. This is where sperm's produced. The Leydig cells will also be kind of tucked in here, and these guys will be responsible for hormone production like testosterone. The scrotum is there because you need to make sure that the testes, where the sperm's being created and where it's maturing, is about 2 degrees cooler than body temperature. And so by being able to move it away from your core or pull them up towards your core if it gets very cold, you can try to regulate that so it doesn't cause lowered sperm count. This is also why you shouldn't wear whitey tighties if you want to have a high sperm count or look okay. The epididymis is these regions here that you see around the edge that are a little bit more sac-like. That's where the sperm, after they're produced, they have to kind of mature, where they get their tail properly, they get all the internals proper. And so that's the part where they would just kind of rest and mature for probably like, a day or so would be what's required. And then from there, they can leave via the vas deferens. So that's the tube that kind of comes up through here and eventually dumps into the urethra. That's this part right here. The urethra itself will go eventually through the penis, and that's where you will excrete both urinary waste, because this is your bladder here. Your bladder also drains into this, as well as you would release semen. You have semen production itself is going to be done by a bunch of these glands that you see. That's the prostate there. You've got the Cowper's gland. You've got seminal vesicles. But you don't have to know their names so much, but there's a series of different glands that will secrete things like nutrients, that will secrete different things that are coagulants or anticoagulants to help control the viscosity, if you will. They secrete buffers and such, so the pH balance within the female reproductive tract doesn't kill the sperm. So they'll add a bunch of stuff. It's not like it's just sperm that you'll have in actual semen. There's a bunch of other stuff. And then, as we said before, you have the urethra, and the penis itself will be the external structure where everything ultimately is released from. Now, the female anatomy, you're going to see it's very similar. Still digestive tract. Obviously, that's unchanged. The big difference is you've got the bladder here with the urethra, and they are separate from the reproductive tract. And so you're going to have the ovaries. You're going to have the oviducts or fallopian tubes that then lead into the uterus. You have the uterus, this muscular organ where the baby would essentially be grown. I don't know what you're going to say. And then you've got the cervix, which is where you kind of mix, where the uterus dumps into the vaginal tract or the birth canal. And then you've got the actual vagina here, which will be the one that will exit. Once again, notice you've got 3 separate exits, and notice their close proximity, which can lead to issues. Males aren't as prone to this because all their stuff's kept much more separate, so they don't have the increased likelihood of contamination by bacteria from other areas that are waste areas into either the urinary tract or the reproductive tract. And then you'll have the external structures, the clitoris, and then the labia. Those will ultimately just be where everything physically dumps out into. Females don't have any dramatically exterior structures like males do. The actual labia, the clitoris, and all that stuff, you'll see oftentimes that's referred to as the vulva. That's just the sum total of all the exterior stuff within females. The clitoris is really only notable because it's the only organ known to only provide pleasure functions. So you can decide if that's a good or bad thing, but that's its claim to fame. We'll discuss the corpus luteum later, but that will be a structure that will typically hang out by the ovaries, and it'll help produce hormones. So beyond that, that's a quick overview of the female reproductive system. We'll get the follicle later. That's going to be what develops into the ovum. Now, spermatogenesis. With guys, when they go through and hit puberty, they're going to start taking these germ cells that will produce a bunch of other cells. All right, they're called spermatocytes. And so some of these cells they produce that will become spermatocytes will go through meiosis called spermatogenesis, and they'll produce 4 sperm cells. Now, obviously, these sperm cells will be haploid because that's what meiosis does. But the basic idea here is that you start out with 1 2-end cell and you end up with 4 1-end cells. This works because sperm are small, they're supposed to have minimal investment, and so you don't have to worry about trying to, consolidate all your stuff in one cell. We can make 4 small sperm cells and no one's worried about it. That's fine. That's the male strategy. Now, the female strategy, which is going to start, once again, really around puberty, is where you're going to start finishing this process. That's when you start to, end this. But really, when you're a fetus, you're actually going to produce all the follicles, which are what can go through this process of OO or oogenesis once you hit puberty. So this process starts earlier than in males. It just kind of freezes in meiosis 1 until you actually hit puberty. And then this process continues till you're probably about 35 to 55 when you hit menopause, at which point it stops, period. In males, notice it won't stop. And then this process will result in just one ovum. So you start this process with one 2N cell, but it's not like spermatogenesis where I got 4 N cells. That's not going to happen. Because the ovum has to have most of the organelles, it has to have the cytoplasm, what you're going to do is whenever you divide, you're going to kind of cheat one of the cells when you divide. And so that cell has very little cytoplasm, not enough to make it. So we'll call that a polar body, and that one will just die and be reabsorbed. And then ultimately then you'll end up with, boom, one ultimate ovum. This makes sense because when things like humans reproduce, which typically are doing one set of meiosis, one time of doing oogenesis, you end up with one kid. We're not having litters here. And there's a reason that we're not ending up with 4 kids in general. We can have less. All right, the female cycle. I know everybody loves discussing this. So with females, there's actually 2 things going on. You're going to have either the estrus or the menstrual cycle. Estrus just means that you reabsorb most of the endometrium, the lining of the uterus that you've produced during what would be considered like the menstrual cycle. Whereas with the menstrual cycle, which you find just in like us and some primates, you're actually going to slough off most of this endometrium and it's going to leave the body. And so you'll have a lot more blood and tissue loss because of this. Now, it doesn't make quite as much sense in one sense as the estrus cycle because you could have tried to reabsorb a lot of those nutrients, but it's how we do. You'll also have, usually coinciding with the menstrual cycle, the ovarian cycle. And so this is going to be about growing a follicle to get it to the point that it will ovulate, which means essentially spit out an ovum. The ovum then can go through fertilization, can become a zygote, and you have a baby. If this process doesn't work, if you go through and you don't end up with fertilization, etc., you'd go back into a normal menstrual cycle and the whole thing would start again. Ovarian cycle, you're going to start maturing another follicle, which will eventually give you another ovum, and the process will continue until a pregnancy results to kind of temporarily halt all this. Now, more detail. Once this follicle starts to go through and develop, and that's going to occur because of a jump in FSH, follicle stimulating hormone. So that's going to start this follicle growing in size. It's going to cause it to kind of go through the process of meiosis and ultimately produce this ovum. Now, ovulation, the release of the ovum from this follicle, is going to occur because of a spike primarily in luteinizing hormone, LH. You'll see there's a small one of SSH, but the bigger deal usually is this LH spike, so that's its purpose. And once this ovum is released, it's going to go into the ovary, which is this structure, and then through the oviduct where fertilization may or may not happen. The rest of this structure stays behind the rest of the follicle and produces hormones, like progesterone, to help make sure that the uterus stays prepped, because during this process you have a separate thing going on, which is going to be the uterine or menstrual cycle, where at the start of this process, you'll see this whole process starts with the beginning of essentially a period. So you're going to void all the endometrium, and then you're going to start producing the endometrium, and that'll be triggered by this rise in estrogen that you see going on here. That rise in estrogen also can trigger this LH spike, so you know to ovulate. And then after that, you're going to get this corpus luteum, this leftover chunk of the follicle that's luteum means yellow, so it's yellowish, you can see it literally is. That'll start producing progesterone, which will help maintain this endometrium, this lining of the uterus, so that implantation can occur if fertilization has occurred of the ovum. Now, if there is no implantation, then ultimately the corpus luteum will degrade. This is actually a remnant of an older one. It's called now corpus albicans because it turns white as it kind of shrivels up. And so if this process doesn't occur, if the embedding doesn't occur to start secreting its own hormones, to kind of say, hey, we're good, let's keep going, if that doesn't happen, then this corpus luteum tends to kind of fade away, and so you'll see the levels of progesterone drop off, and then the whole process just resets and takes another stab at it. You're just going to go through another set of things. It is important to realize, though, the ovarian cycle, while it's closely tied to the uterine cycle, they're not exactly the same. And so this is why they'll say you have to worry about the fact that you're not always going to ovulate at the same part of your uterine cycle. So if you're trying to say that I'm going by ultimately when a period is and trying to count days to try to avoid sex on certain days to avoid getting pregnant, that's not a guarantee. That's a decent way to get a baby because you can have where ovulation occurs over a wide spectrum. It's usually about 13 to 15 days after the start of your period, but it can be 5 days after. It's not an exact thing because your hormones can kind of fluctuate differently from each other. Because you've got your estrogen and progesterone that are kind of doing their thing, and you've got your FSH and your LH that are doing their thing, and they might exert some influence on one another, but it's not perfect. The best guess we have for ovulation is actually a spike in body temperature, but even that's pretty small. You're talking, maybe half a degree. And your body temperature typically stays up there, so you really got to catch it immediately when it happens. So there are ways with which you can try to kind of figure out exactly when you're ovulating, but they tend not to be perfect, and you have to have a fair amount of equipment and really keep an eye on it. Otherwise, it's best just not to count on that. Now, with pregnancy, there are some people that think orgasms, this rhythmic contraction of the reproductive muscles, might help cause pregnancy, especially with females. There's some idea that maybe it would help draw semen in, but there's pretty good evidence, I think, saying that that probably is not completely the case, if at all the case. So we're not always positive exactly in females, at least, what this is going to accomplish if there's other than just, I guess, fun, if there's a really good use for this as far as causing pregnancy. Fertilization or conception is just going to be that moment in the oviduct that the sperm meets the ovum. And then after that, several days after that, you'll have where the zygote, that first fertilized cell, will start to undergo cleavage, which is splitting, and then later it will implant in the actual uterus, the endometrium, but at that point it'll probably be like 8 or 16 cells. So it doesn't do so immediately. There's actually several days within which it travels through the oviduct, or fallopian tube if you want to call it that, before it implants. And if it implants, it starts to produce this HCG, human chorionic gonadotropin, so it's a specific hormone that kind of says, hey, I'm here. And so it triggers the corpus luteum to keep producing progesterone, to keep these hormone levels up, so the process doesn't reset itself. So this is basically what says, hey, I'm pregnant. This is also what pregnancy tests are checking for. They're looking for levels of HCG, which if they're spiked, tend to indicate, hey, I'm preggers. All right? Now, if you want to avoid getting preggers, you can go through and use contraception. Now, there's 3 basic ways that contraception avoids you becoming pregnant. The first is no sperm or no egg, period. So if you go through and you have certain types of birth control, they prevent ovulation. If there's no ovum, sperm has nothing to fertilize. No baby. You can also go through surgical means, either tubal ligation in female where they mess up your oviducts, so that way even if you go through ovulation, the ovum will never reach the sperm because there's a gap. You've got like the old railroad bridges and it's like you blew up the middle. The same thing happens with males with a vasectomy, where they do the same thing to the vas deferens. And so that means that sperm cannot actually get through the vas deferens to make it to the urethra. You will still have the seminal secretions, so you'd still have semen, but it would be minus the sperm. And so at that point, no sperm, no babies. You can keep the sperm and eggs apart, so this would be like condoms, diaphragms, any barrier that's just going to prevent the sperm from reaching the egg physically. You can have coitus interruptus, where the goal is the pull-out method, where if you don't have the semen actually inside of the female reproductive tract, that nothing will happen. That one's not particularly effective, so you might want to not trust that too much. And you have the rhythm method, which is you avoid sex during the times with which the female is most likely ovulating. If you're really, 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 really good at trying to do this, it can be like, 95, 97% effective. In practice, it's more like 80% effective in a year, meaning that 20% of people in any given year of doing this or more will end up with a baby. So not the best. Whereas birth control tends to be 99-point-something% effective. Surgery tends to be 99-point-something% effective. And then lastly, you can prevent the actual implantation. So even if you do have a zygote or an embryo at that point, technically, it can't implant. And so you've got intrauterine devices will do this, and some forms of birth control can do things like increasing the mucus that's there, so it makes it harder for the actual embryo to embed and start to grow the blood vessels, etc., needed to develop a placenta and to properly implant. And so these are very effective, but these are also some of the more controversial methods because you did technically get conception. You're just preventing it from actually becoming a pregnancy. Now, if something appears to have gone wrong during a pregnancy, and this can be detected using the simple non-invasive types like ultrasound. So if they have reason to suspect something could be problematic, there are some ways they can do tests. And the invasive common ways they can do it is if you're later along, like say probably 16 to 22 weeks, they can take a large needle and they can essentially stab it into the female, into the amniotic fluid that surrounds the baby, that's the sac that surrounds it, and they can take some of that fluid, which contains cells that the baby sloughed off. They can then genetically test those cells to find out if the baby has Down syndrome or a variety of other disorders. If it's earlier than 15 weeks, usually about 8 to 15 weeks, they can also do chorionic villi sampling, CVS. And for that one, they normally just go in via the vaginally, and they can go through and they can go into the uterus and they can take some parts of the placenta. The placenta oftentimes have these little extensions, and so they can just kind of suction or snip off some of those. And because those have the DNA of the baby, they can then do genetic testing and also find out if there's something wrong. Now, the problem with this for some people is if you do this testing, what do you do about it? I mean, there is like a 1% risk that it could cause miscarriage, so there's some risk to the child, I suppose. But even beyond that, if you find out the child does have something genetically wrong with it, that the child would be disabled, the child could have disease, maybe the child wouldn't live very long, then there's some stuff of, well, would you abort the child? Would you just prepare? What do you do in response to that? And so there are ethical questions, and some people would try to avoid this because they don't want to have to deal with that. They just wait. This happens to be some of the chorionic villi type stuff that they try to pull off. That's just what this is. And this is an ultrasound. That's the baby's head, legs, arms, I believe, torso. You'll notice its head's ginormous. That's usual at that point. And the last bit on the cover for this one is infertility, especially as people wait longer and longer due to birth control in many cases to have children. There's not as many surprises. So because of that, there's more and more issues with older mothers and infertility, where they have difficulty getting pregnant. And so there's several ways to try to get around this. The first and cheaper way is to take a medication that makes you ovulate a bunch. So Clomid's the common one there, where it basically just tells your body, like, let's do oogenesis. Let's do it 2 more times for fun. And so the downside is if you do have kids, you can oftentimes have a litter of kids. So you might have twins or triplets or like 6 kids, like John and Kate plus 8 or whatever that was. They ultimately were on drugs to make that happen. So it's not quite as immaculate here or amazing. It's the drugs. Now, if that doesn't work, you can also do in vitro fertilization. And this is where they'll take eggs, where they'll literally like go in with laparoscopic stuff, go into the ovary after you took medication to make a bunch of ovums be created. They'll then pull out the eggs. They can then mix them with sperm in like a petri dish, what they used to call test tube babies, although I believe it's a petri dish. So that way you get fertilization there. They can then grow them in the dish till they get to about like an 8-cell stage, and then they can take and go to the mother or the surrogate or whoever who's been taking hormone, progesterone, to make sure that the uterus is perfectly ready, and they literally just kind of go in there vaginally and shoot out the embryos into the uterus so they can implant. And so this is one that goes an extra step. This is also what you'll see with many surrogates nowadays. They're gestational surrogates, and they'll do a process like this, where somebody who donates an egg, somebody who donates sperm, they then take it and they'll put it into the surrogate so it's not genetically hers. That way it's not quite as weird when you're like, here's your baby, because it's typically not your baby genetically.